in these short talks to contact those vendors and exhibitors directly, and they'll be happy to answer all your questions and go into as much detail as you'd like. So the first up will be Biognosys. Biognosys is a Swiss company, so thank you for sharing your evening with us, Fabia. We are going to hear about an application based on their targeted and DIA workflows. This will involve IRT kit and what I hope I'm pronouncing correctly as Quick C3. So Fabia, Simona will give this talk, and again, there will be brief time for questions, and then we'll move on. So over to you, Fabia. Thank you.
Thank you very much, David. Can you hear me?
Yes.
Okay, can you, yeah. Okay. Thank you very much for the introduction. My name is Fabia. I'm a technical sales manager at Biognosys. And today I will present two products that we wanted to bring to you as Supo and that we still launched in March. The new IRT kit and Quick 3. What we hope it is already or will become your dream team for quality control. First off, I would like to say a few words about our company. The core of Biognosys resides in a leading R&D department, which continuously develops know-how that is then transferred to contract research services as we support projects with our pharmaceutical and biotech, but also academic partners through contract research services, as I said. On the other side, we develop products, specifically software and kits for mass spectrometry. The success of our company resides into the continuous and direct interaction between our three departments. Biognosys was founded in 2008 as a spin-off company from the group of Professor Rudi Ebersold at the ETH in Zurich. And in 2012, together with the ETH and SciEx, we co-developed DIA, also known as SWOT or HRM mass spectrometry. In the same year, we also started an early distribution of our IRT kit. In 2013, then, we started developing the first software packages for quantitative proteomics as well. So our development is a journey that expands towards new technologies and applications. In 2018, we established a drug target deconvolution platform, and at the moment, we are in the process of implementing a GCP laboratory for clinical samples. In this way, we can provide cutting-edge services and products to over 250 partners so far. Last year, we have supplied to the community over 3,000 software and kits. Over 2,000 of those alone were IRT kits. And indeed today, I will talk about our best seller, the IRT kit, or standard peptides for indexed retention time. The IRT kit contains 11 stable, non-naturally occurring peptides that evenly spread over the retention time space, and they function as an anchor point for retention time prediction for any other peptide contained in a sample. Indeed, although a peptide retention time is determined by its physicochemical properties, one also has to account for systematic variability based on chromatography and metrics, and this can actually account for a large shift in retention time. Small shifts are also caused by unpredictable fluctuation from injection to injection. The IRT kit addresses this first challenge as it enables retention time normalization and as such comparability across instruments and acquisition methods over time. And this is why spiking the IRT kit in your runs is a robust solution for LC-MS QC. Obviously, using the IRT kit also benefits the accurate quantification during targeted data extraction. So our new IoT kit contains the same peptides, which means there is no need to adjust your current workflow. However, you can now count on a more balanced intensity of peptide signals, which minimizes peptide undetectability even under suboptimal LC conditions. It makes it easier to monitor different parts of the gradient. The IRT kit peptides are now quantified by amino acid analysis, which provides accurate information on peptide amounts and fully enables the use of the IRT kit for quantitative quality control as well. Of course, this also improves long-term reproducibility as we have minimized the batch effect across IRT kit packages. So as we all know, the quality of any conclusion from proteomics data critically depends on the quality of the raw data, and having the same set of peptides spiked across runs is a robust and accurate QC solution. We have co-developed our IRT kit together with our software for QC, Quick. Quick is a free software that allows for instant control of the most important instrument parameters. So what is new in Quick? Threshold controls. You can set individual thresholds for all instrument parameters. In this plot, we have set thresholds for identified precursors and protein groups. And the thresholds are visualized as green windows. And basically, every run that falls off the window is marked as an outlier. You also have the opportunity to set and visualize event tags. So for instance, if you acquire data right after column replacement or system recalibration or lens cleaning, any event in your facility that might have an effect on instrument performance can be tracked. Improved scalability. You can monitor the performance of more instruments, and this is also allowed by the fact that Quick has a significantly reduced analysis time if you compare the last release, Quick 3, with the previous generations of Quick. So we are in the range of just a bit more of 100 seconds for a DDA run with a 20-minute gradient using a C. elegans spectral library as background, and 155 seconds for a DDA run with a 120-minute gradient on a HeLa spectral library. Besides improving our spectral library support, we have also introduced support for FASTA files that can now be used as background proteomes. And so with this, I hope that you are ready to benefit from our dream team for QC. If you want to request your IoT kit, or if you want to give it a try to Quick, please just request your Quick free license. Either you can visit our shop or write us at sales@biognosys.com. And obviously, if you have any other question regarding our products and or services, please do not hesitate to contact us. Thank you very much.
Okay, thank you, Fabia. A quick question that occurred to me on your Quick application. Does that use sort of native instrument file formats, or is it some extracted, open source?
So Quick is compatible with vendor file formats. We support at the moment Agilent, Thermo, Bruker, Waters. So yeah, you can use your run files.
Okay, and a question that's come through from the audience is, Quick 3 compatible with older IRT kits?
No, so as I said, we co-developed Quick to function, to work based on our IRT kit peptides. So the IRT kit is necessary in order to retrieve QC information with Quick.
Okay. And a question also on the IRT kit. How about hydrophobic peptides, which tend to be lost in chromatography? Is this better with the new kit?
So yeah, we should be able to better cover the gradient thanks to a more stable signal intensity. Of course, the end part of the gradient is more susceptible to be unstable, so it might still be a bit of an issue, but I think it makes sense to try, and again, I mean, I think there is really specific cases, and for sure this person can get in contact with us for more specifications about this particular situation, but yes, we should be able to cover the gradient.
Okay, great. So what will probably be the last quick question comes from Birgit, who asks, how do you make the batch-to-batch reproducibility better, as you had mentioned? I guess what are the factors there?
Yeah, this is given by the fact that now we can quantify by amino acid analysis. So we now have complete control on our peptide mixture. So this is something that we were not guaranteeing before, and we would not recommend using our IRT kit for quantitative quality control, but only for retention time normalization or retention time quality control. But given the fact that now we have a production certification on our on the quantity that we produce, we can now guarantee that there is no batch-to-batch variability.
Okay, very good. Thank you. And thank you to Biognosys for that presentation. So we'll move on now to the next short talk. This is from Resin Biosciences. And Resin enables or enhances proteomic sample preparation with a portfolio of enrichment and affinity capture media. So today we have Stoyan Stoichev, who will tell us, and I apologize if I butchered your name, will tell us about high-capacity magnetic microparticles.
And thank you for the opportunity to present. Just start sharing my screen. Okay. So my name is Stoyan, and I work for Resin Biosciences, focusing on developing some of the proteomics applications in the company. So today I would like to give you an overview of our technology and how we are applying this to improve the efficiency and automate various workflows, specifically in this presentation when it comes to proteomics. So as you all know, automation brings several benefits with it, including time and cost saving, increases in throughput, as well as higher reproducibility, and therefore ultimately better data quality. Magnetics allow for these processes to be simplified. So for example, we don't have the need to centrifuge samples, nor to have additional equipment such as vacuum manifolds and so on. A rather simple and inexpensive bottom pull down or pull up magnetic system, such as the one shown in the middle here, all that is required. Magnetic workflows are also very easy to scale. So as opposed to solid phase extraction cartridges, where one needs to match the cartridge size to the input material, with magnetics we are just decreasing or increasing the volume of beads based on the application requirements. Our mag resin range is also engineered for efficient handling and automation. We have patented polymer technology that provides significant increases in